Hi, my name is Sylvia Diaz and I'm from Garden City, New York. My research project is based on a mutation within the cassava plant that I believe makes it resistant to a devastating virus called CMD. But first, why is the cassava plant so important? Currently, changes in temperature and precipitation have negatively impacted the production of crops. However, the cassava plant provides hope, as recent studies have shown it's resistant to climate change. Not only this, but the cassava plant is a staple crop for 700 million people in mainly third world countries. And so the highly infectious virus, CMD, should be taken very seriously as it impacts millions of lives. And so, I've been inspired to work with the St. Louis Stanford Plant Science Center in order to fight this virus. In my research project, I hypothesized that the G680V DNA polymerase delta mutation is a source of resistance to the CMD virus. This is because the mutation is within the fingers, and so it could impact the overall structure of the DNA polymerase. In order to confirm this, I used Swiss Malder to construct a homology model of the mutated cassava plant using the yeast DNA polymerase. With the mutated cassava, I was able to create electrostatic surface potential diagrams. Upon close examination, I noticed that the mutated cassava has an increased narrowing in the DNA binding region. And so I was able to confirm that the mutation impacted the overall structure of the DNA polymerase. My research is based on a glycine to valine mutation. Both of these molecules are nonpolar amino acids, but the larger valine within the mutated cassava causes the surrounding amino acids to reorient. I illustrated this by performing visual molecular dynamics, and in this image, you can see in the mutated cassava there is a yellow molecule called argon 681 that is in a different orientation. This is of key interest because it demonstrates that the G680V mutation may be impacting the DNA polymerase and the CMD virus's interactions. Therefore, I measured the relative distance between argon 681 and the surrounding amino acids like aspartic acid 665 and glutamic acid 524. This graph illustrates the mutation causes the surrounding amino acids to lose attraction, with the large spikes indicating the breakage of salt bridges. And so, all of my data indicates that the G680V mutation inhibits the replication of the CMD virus, making the cassava plant resistant. Now, although I've accomplished a lot, I'm continuing my research by running molecular dynamic simulations through Stony Brook University's supercomputer called Seawolf. With this software, I'm able to observe in nanoseconds the conformational changes between protein and DNA. I truly believe this mutation should be incorporated in all of the cassava plant's population, and so that this is a new adaptation for survival.